opportunity to speak and address this issue. On the proposed no significant risk level for the chemical glyphosate to be adopted into regulation in Title 27. I first say that there are no safe levels of glyphosate. That statement originated with me in my studies over the past several years. I've written 6 papers on the subject. I have 2 more in process. I'm continuing research in glyphosate. Glyphosate is a synthetic amino acid and analog of our canonical amino acid glycine. It participates in both plant and animal biology, contrary to what was originally thought by Monsanto, that it only affected plants, archaea, and bacteria. 1 microgram of glyphosate technical acid, N-phosphonamethylglycine, contains 3.561 trillion molecules. Each of those molecules are capable of integrating with protein, altering shape, folding, and function. I am a U.S. scientist and hazardous chemical material consultant and expert on the subject of glyphosate, and one of the few people that received all of Monsanto's trade secret studies of glyphosate from the U.S. EPA. The federal agency supplied me these documents in excess of 100,000 pages. I now have 6 peer-reviewed papers. I call on the California Environmental Protection Agency to immediately ban this chemical and not to set a limit of 1,100 parts per billion for this chemical. There should be 0 tolerance for this chemical, and I'll explain. Monsanto claims glyphosate to be safe to animals and humans because they do not possess the shikimate pathway, a pathway which is disrupted by glyphosate in plants, archaea, and bacteria. Glyphosate is known to disrupt the enzyme EPSP synthase, or 5-enolpyruvyl shikimate triphosphate synthase. However, this statement is now inherently false, as glyphosate was found by SAMSEL in 2016 to inhibit digestive enzymes and others found in animals and humans. Glyphosate is a protease inhibitor, including digestive enzymes. Such disruption of human enzymes is well known to lead to a host of modern diseases, including cancer. Again, lysosome is an antibacterial enzyme that is an integral part of the innate immune system of humans and other animals. Monsanto found significant tissue damage to all glands and organs in their 2-year long-term studies of glyphosate in mice and rats. Tissue damage stimulates the production of fibrocytes. Glyphosate reaches the end of the line in the capillaries in the extracellular matrix, where it is escorted 1 molecule at a time into the cell, where it participates in protein synthesis and is excreted by the cell. This I noted in fibroblasts also produce the structural proteins, which include the 27 types of collagen, elastin, glycosamine glycans, and the glycoproteins of the extracellular matrix. So glyphosate is along for the ride, even bridging assembling strands of proteins affecting shape, folding, and function. Glyphosate should not be part of any biology. Also, fibrocytes and fibroblasts are differing states of the same cell, the fibroblasts of which are involved in immune regulation via TAF-derived elements of the ECM modulators. These ECM components, like TSP1, are associated with sites of chronic inflammation and carcinomas. This is where glyphosate causes many funky cancers, as its association with fibroblasts. I've published results of lab analysis and experiments with glyphosate integration with structure. Stephanie Seneff, my colleague, is also co-author on all of these papers with me.